Welcome back to the deep dive. As medical microbiologists, we tend to focus on, microbes that make us sick.
Makes sense, right?
Yeah, definitely.
Yeah.
But today, we're going to dive into something a little different, extremophiles.
Now, these guys are fascinating. They thrive in conditions that would kill most other organisms.
Exactly. And that's why we're taking a deep dive today. We're looking at Volume 5 of the second edition of Bergey's Manual of Systematic Bacteriology.
Ah, the Bible of bacterial taxonomy.
Pretty much. And we are honing in on a particular group within the phylum Actinobacteria, specifically the class Thermoleophilia.
Now, I bet you can guess from the name what makes these bacteria special.
Heat lovers.
You got it. They thrive in scorching hot environments.
Makes you wonder how they do it, right? What kind of adaptations they have.
Well, that's what we are here to uncover. And trust me, their survival strategies are pretty remarkable.
So let's start with the basics. This Thermoleophilia class, it's a relatively new classification, isn't it?
Yeah, it is. Originally they were grouped under Rubrobacterialis.
Right.
But based on 16S RNA gene sequencing and phenotypic properties, they earned their own class.
And within this class, we have the order Thermoleophiles.
Named after the type genus Thermoleophyllum. And I think the name says it all.
Heat and oil loving.
Exactly. Thermoleophyllum are these tiny little rod-shaped bacteria.
Gram-positive, I'm assuming.
You bet. Non-modal, too. But here's where things get really interesting, especially for a medical microbiologist like yourself. Instead of sugars like glucose, these bacteria prefer something a little more unusual.
Let me guess. It's not the usual bacterial fare.
Not at all. They prefer alkanes as their main source of carbon.
And alkanes. Hydrocarbons.
Precisely. It's like they're running on oil instead of sugar. Pretty wild, huh?
It is. I mean, most of the pathogens we deal with, they're all about that glucose.
Right. But these guys, they've got a different strategy. And it's this kind of unique metabolism that makes them potentially valuable for things like bioremediation.
Bioremediation. So using these microbes to clean up pollutants.
Exactly. Think oil spills, for example. Thermoleophyllum, with their alkane-eating abilities, could be real game changers.
I see. So they're not just fascinating from a scientific perspective. They might have real-world applications too.
Absolutely. Their unique metabolism could hold clues for developing new antimicrobial strategies or even understanding antibiotic resistance mechanisms.
Now, given their name, Thermoleophilia, I'm guessing they thrive in pretty extreme temperatures.
Oh yeah, we are talking about temperatures that would kill most bacteria, like 55 to 62 degrees Celsius.
Wow. And they need oxygen to survive, right? They're obligate aerobes.
They are. But here is something that threw me for a loop. Increasing the oxygen levels doesn't necessarily make them grow faster.
Really? That's surprising. Most bacteria, the more oxygen they get, the happier they are, the faster they grow.
Right, but Thermoleophyllum have a unique respiratory kinome, MK7H4. It's like they have a different playbook for handling oxygen.
So that's how they adapt to these oxygen-rich environments.
Fascinating. And speaking of adaptations, I was reading that Thermoleophyllum doesn't seem to rely on the tricarboxylic acid cycle, the TCA cycle, for energy.
That's a big one. The TCA cycle is central to energy production in so many organisms, including a lot of the pathogens we deal with every day.
So how do they do it? How do they generate energy without the TCA cycle?
Well, they have the glyoxylate cycle. That seems to be their workaround, their alternative pathway.
So while other bacteria are relying on the TCA cycle, Thermoleophyllum has found a different way, and that difference could be crucial, right?
Exactly. Especially if we're thinking about new ways to target bacterial metabolism.
Now I want to talk about the elephant in the room, or rather the unusual structures in the room.
I know what you mean.
Figure 425 in Bergey's shows these interesting formations within the cells.
Yeah, those are some curious structures.
They almost look like little compartments. What's the thinking on those?
The current theory is that they are involved in alkane uptake and storage. So it's like their specialized fuel tank for those hydrocarbons.
Exactly. And these internal structures are one of the things that set Thermoleophyllum apart from other thermophilic bacteria, like Thermus and Thermomicrobium.
And Table 318 in Bergey's nicely lays out some of those key differences, right?
Oh, yeah. Cell wall composition, respiratory kinones, preferred carbon sources, it's all there.
It highlights just how diverse these bacteria are, even when they share a love for heat.
Absolutely. But there's also a challenge with these bacteria.
Oh.
Culturing them. They are notoriously picky eaters, and even under ideal conditions, their growth is pretty sparse.
So studying them in the lab can be a real headache.
Yeah, it can. But it also makes you wonder how many other fascinating microbes are out there that we haven't even been able to culture yet.
All right, so we've covered a lot of ground on Thermoleophilia, but let's see how much has really sunk in. Time for a little pop quiz. Are you ready?
Hit me with your best shot.
Okay, first question. What is the primary carbon source that Thermoleophilia species utilize? And how does this differ from many human pathogens?
I think our listeners can handle that one.
All right, question 2. What's unusual about Thermoleophilia's response to increased oxygen levels? And what unique respiratory component plays a role in this?
Think back to what we discussed about their adaptation to those oxygen-rich environments.
Question 3. Why is the apparent absence of a complete TCA cycle in Thermoleophilia significant, especially from a medical microbiology standpoint?
Remember that the TCA cycle is like the powerhouse for many bacteria, including those that cause disease.
Question 4. What internal structures are observed in Thermoleophilia and what's their potential function?
Picture those unusual formations we saw in Figure 425.
And finally, question 5. Name at least 3 characteristics that differentiate Thermoleophilia from other gram-positive thermophilic bacteria, like Thermus and Thermomicrobium.
Remember, Table 318 is your friend. Take a look and see if you can spot the differences.
All right, those are your 5 questions. Take a moment to ponder those, and we will be back in a flash to break them down together.
Looking forward to it.
Okay, let's go over those questions and see how well you did. Don't worry, this is all about learning and exploring.
Exactly.
No pressure.
All right, the first one, the primary carbon source for Thermoleophilia. What was it again?
N-alkenes, those hydrocarbons we were talking about. Remember how he said it was like they were running on oil?
Oh, yeah, that was pretty wild. Most bacteria we think about in medicine are all about those sugars, right?
Yeah, they love that glucose. But not our Thermoleophilia, and that really highlights the incredible diversity of bacterial metabolism, doesn't it?
It does. And it makes you wonder what other unusual metabolic pathways might be out there.
Right. I mean, we've only scratched the surface of the microbial world.
Okay, question number 2. Thermoleophilium's response to increased oxygen levels. What was so unusual about that?
Well, unlike most bacteria that thrive with more oxygen, Thermoleophilium's growth isn't really affected.
Yeah, it didn't really boost their growth. Right. And the key to that is that unique respiratory quinone, that MK7H4.
MK7H4, got it.
Yeah, it's like they've got their own special way of handling oxygen.
And that could be really important when you think about how bacteria adapt to different environments, right? Like even within the human body, you have areas with different oxygen levels.
Absolutely. The human body is like a whole world of different microenvironments.
All right, moving on to question 3. Why is that missing TCA cycle so significant in Thermoleophilia, especially for us in the medical field?
Well, you have to remember that the TCA cycle, it's this fundamental metabolic pathway.
Essential, right.
Yeah, for a lot of organisms. I mean, think about pathogens like E. coli or Staphylococcus aureus. They rely heavily on the TCA cycle for energy.
So it's kind of like their engine, right? Their metabolic engine.
Exactly. So the fact that Thermoleophilia can function without it, it's pretty remarkable.
They're going off-roading.
That's a good way to put it. And they are probably using that glyoxylate cycle instead as their alternative route.
It makes you think, could there be other bacterial pathogens out there with similar workarounds? I mean, we know there are tons of bacteria we haven't even identified yet.
Oh, absolutely. And that's what makes studying these extremophiles so important. They can give us insights into these alternative pathways, which could even lead to new ways to target bacterial infections.
That's a really cool thought. All right, question 4. Those strange structures we saw inside Thermoleophilia, any ideas on their function?
Those are most likely involved in alkane uptake and storage. You know, they need a way to deal with all those hydrocarbons they're consuming.
It's like their specialized fuel tank.
You got it. A perfect adaptation to their hydrocarbon-rich diet.
And last but not least, question 5. Those 3 key characteristics that set Thermoleophilia apart from Thermus and Thermomicrobium, you know, those other gram-positive thermophiles.
Well, first off, there is that main carbon source. Thermoleophilia uses those N-alkanes, while Thermus and Thermomicrobium don't.
Right. And then there was that difference in their cell walls.
Yeah, Thermoleophilia have diaminopimelic acid in their cell walls, whereas Thermus has ornithine.
I remember that now. And of course, we can't forget about that unique respiratory quinone, that MK7H4 that Thermoleophilia has.
Right, that was a big one. And all of those differences, they show us how even though these bacteria all love the heat, they've evolved these distinct adaptations to thrive in their specific environments.
So even among heat lovers, there's diversity.
Exactly.
Well, that wraps up our little quiz on Thermoleophyllum and the class Thermoleophilia. Hopefully, this deep dive into Volume 5 of Bergey's has given you a new appreciation for these amazing extremophiles. It really makes you think, these extremophiles, like living in these crazy environments, using these weird energy sources, it challenges our understanding of how life, how life even works.
It definitely, it forces us to reconsider what we think of as habitable, like for us, boiling hot springs or oil-contaminated soil. Those seem pretty extreme, but for these microbes...
An home sweet home.
Exactly, and it makes you wonder, are there similar adaptations happening in other bacteria? Maybe even those that could impact, human health.
Right. If they can thrive in those conditions, what's stopping them from adapting to, say the acidic environment of the stomach?
That's a really good point. The human body, it has all these different microenvironments, the gut, the skin, even different parts of the respiratory tract.
They all have their own unique challenges for microbes, right? Like different pH levels, oxygen availability, all that stuff.
Exactly. And studying extremophiles like Thermoleophyllum can give us clues about how bacteria might adapt to those challenges.
So it's not just about understanding these weird microbes living in extreme environments. It could actually help us understand how bacteria evolved to survive in the human body.
Absolutely. And that has huge implications for things like antibiotic resistance.
Because if we can understand how they adapt to stress, like, say, the stress of being exposed to an antibiotic,
then we might be able to develop new strategies to combat resistance.
it's all connected, isn't it?
Yeah.
The more we learn about these extremophiles, the more we learn about the microbial world as a whole.
And the more we can apply that knowledge to human health.
It's pretty amazing when you think about it.
It is. It just shows that there is still so much out there to discover and learn about these incredible microorganisms.
Well, I want to thank you for taking us on this deep dive into Thermoleophyllum and the class Thermoleophilia. It's been fascinating.
My pleasure. Always happy to talk about these amazing bacteria.
And to our listeners out there, thank you for joining us on the deep dive. We hope you enjoyed this exploration of Volume 5 of Bergey's and learned something new about the fascinating world of extremophiles. Keep those minds curious, and we'll catch you next time.